First of all, I would like to thank the organizer for giving me the opportunity to speak today. The topic I will be discussing involves the genetic probes technologies and 3D electron microscopy applied to obtain nanoscale maps of all cells and tissue. A common challenge in biomedical research is to be able to observe and map biomolecules in the intercellular and tissue context. And this is particularly important in complex organisms where we often lack the knowledge of the function and distribution of many cellular proteins. Our goal at the National Center for Microscopy and Imaging Research, led by Mark Ellisman in collaboration with talented chemists, has been to develop and enhance molecular genetic probes and chemical labeling approaches to facilitate the application of correlated, multi-scale, multimodal imaging to biomedical research, where basically we integrate information across scales, both spatial and temporal, ranging from tissues to cells to specific organelles all the way down to molecular constituents. The expectation is to enable biomedical researchers to follow biomolecules requiring resolution less than 10 nanometers and support the ability to do supramolecular imaging throughout the continuous field of view as great as 100 micron wide. So this range that we refer to as a mesoscale is required to observe and map molecular constituents in their cellular and tissue context. Here is a timeline of the genetic EM probes that we developed in the past 18 years. One key feature of the genetic approach is that they allow incorporation of tags directly onto a target protein of interest, delivering a uniform 3D labeling and providing adequate preservation of the ultrastructures. These, the EM probes you see here have been applied successfully to numerous projects. And more recently, efforts have been made to develop probes that allow detection of protein-protein interactions, such as the HRP method for the extracellular, but also intracellular for split minisog and split apex 2. All these tags induce the oxidation of the aminobenzythine, in some cases induced by light, like in the case of minisog, or by chemically by, like in the case of the apex 2. And this leads to the formation of a polymer that can be rendered EM visible by osmium tetroxide. And in order to achieve the 3D imaging and analysis of biomolecules of interest in their cellular and tissue context, we take advantage of various imaging platforms which can give us specific information. For example, with light microscopy, we can observe the dynamic behavior of proteins via time-lapse imaging. And with the latest advances in the field, that one can push the boundaries in terms of obtainable resolution. By doing correlative microscopy and taking advantage of other platforms like tomography, serial block phase scanning EM, or X-ray, one can navigate across multiple modalities following molecular constituents across scales. And you already heard Eric Bichon talking about examples of application that can integrate these imaging technologies. And I will provide an example of a project where genetic EM probes and 3D EM methods are integrated and implemented. At the center, we use two main 3D EM methods that allow us to reconstruct all cells in tissue. One is the multi-tilt electron tomography, which is a form of transmission EM used to achieve high-resolution 3D volumes of biological specimens. And the other one is the serial block phase scanning EM, or SBEM, which is a volume EM technique that generates large 3D volumes with nanometer resolution. And this has been utilized extensively in neuroscience for 3D visualization of the nervous system ultrastructure and for circuits reconstruction. And in fact, it also provides the 3D architecture of organelles. Now, the wealth of information that we obtain with SBEM from relatively large fields of view is quite remarkable. For example, if you take a sample of the mouse brain, like you're seeing here, this data contain information from all different cell types. This includes neuron organization and functional circuits, the contribution of different glial cells, as well as the vasculature. And because the resolution that we obtain is in the nanoscale, one one can actually interrogate the samples at about various intracellular organelles, but also specific proteins of interest or other non-proteinaceous biomolecules that are critical in many cellular processes. Now, accessing this information has been for a while a bottleneck, with many groups relying on the laborious manual annotation of students who spent a lot of hours to accomplish these tasks. And so more recently, various groups worked to resolve this problem, applying deep neural networks. And our group also found a solution, and this work was led by former postdoc in the lab, Matthias Aberl, and we developed what we call a CDeep3EM. This is a cloud-based tool for image segmentation tasks designed for anisotropic data, such as the ones obtained with the SBEM. This can be launched on Amazon Web Services and can be used for training the deep neural network on 2D and 3D image segmentation tasks. And as you can see here, we applied to various segmentation tasks, including cellular organelles like mitochondria and the plasmic reticulum of an entire Purkinje neuron. Most importantly, this allows us to actually measure specific parameters like the precise volumes, the surface area, the distribution of the organelles. And so we get a whole set of metrics that we can improve our understanding of the role of these organelles and their alterations in disease. These tools allow us to obtain nanoscale maps of all cells and tissue, but one challenge is the ability to differentiate different cell types in complex tissue based on key molecular constituents. And so we work to achieve this goal, and today we can introduce specific EM reporters to differentiate cell types by EM or to target proteins within specific pathways to assess their location and function. And so I'm going to show you an example of such an application. This is a project in collaboration with Tom Nasco group at UCSD, where the goal has been to identify neurotransmitter-defined synapses in a given microcircuits. And to do that, we combine the use of EM probes in transgenic animals, the 3D EMs to achieve high-resolution volumes, and the deep learning automatic segmentation tools to extract valuable information from these volumes. The emphasis of the project was on the structure and function of the synapses, which are specialized connections that are central in our understanding of how the neural network functions. And we focus on chemical neurotransmission, where the neurotransmitters rely on well-characterized vesicular transporter known to catalyze their uptake into synaptic vesicles. We reason that vesicular transporter could represent favorable targets for building tools to image the ultrastructure of neural circuits connections defined by their neurotransmitters. So more specifically, we introduced Minisog tagged to the vesicular glutamate transporter 2 in neurons and to visualize the excitatory synaptic vesicles. And because Minisog is added to the intraluminal loop of VGLU2, the predicted labeling pattern as shown here was a filled dark vesicle. We confirmed the expression at the EM level. As you can see here, the presynaptic terminals are show this intense labeling in the lumen of the vesicles. And to test the probe in vivo, we used transgenic animals and expressed the in-glutamatergic neurons via viral injection into the ventral tegmental area. And using a Minisog antibody, one can look at both at the injection site and the projection sites and observe the labeling of the projecting terminal. We confirmed the VGLU2 was also functional. In fact, it's properly expressed in boutons and colocalized with endogenous VGLU2. And when it's expressed in knockout animals that lack VGLU2, it rescues the postsynaptic glutamate currents. To visualize the label vesicles in 3D, we use the SBEM. And as you can see, the label vesicle can be clearly distinguished within the boutons. But because each presynaptic terminal can contain hundreds or thousands of these vesicles, and in any given area that we analyze, we can have hundreds of them. We apply CDIP3M to automatically detect them. So here is an example of the application of the CDIP3M tool to automatically segment and distinguish the labeled vesicle, which are shown in red, from the unlabeled vesicles, which are shown in blue. And we are working now on visualizing other vesicular transporter in this way, and we started collecting data for VMAT2 for dopaminergic synapses. So we think that these tools will be very useful to the neuroscience community, and will provide for the first time a detailed nanoscale analysis of microcircuits where the identity of presynaptic terminals is specified by their neurotransmitter. To conclude, the combination of these tools allow us to study the morphology of these excitatory synapses in different brain areas. And you're seeing here 3 different projection areas from the VTA, but they also provide us a quantitative analysis of synaptic vesicle profiles. So we can look at their distribution, the size, the density, while we get also information from other organelles, such as the mitochondria, postsynaptic elements, etc. So this would be powerful in the context of many neurological conditions that lead to synaptic dysfunction. So in summary, the capability to combine genetic EM probes and correlative imaging approaches with 3D EM methods with sophisticated computational tools for image analysis has expanded our possibility to visualize biological specimens. And in the future, further development and enhancement continue to push forward our comprehensive understanding of biological functions. I'd like to thank the funding agencies that made this work possible, as well as a wonderful team of scientists at the center, as well as collaborators. And thank you for your attention.